Hello, today we are going to present about cancer detection using microfluidic. Cancer continues to be one of the leading causes of death worldwide. There were approximately 1.6 million newly diagnosed instances of cancer. From the data, you can see that cancer death rate has increased continuously. In order to raise likelihood that cancer patients will recover, more sophisticated technologies are needed to identify cancers before symptoms appear to monitor the progression of the disease and evaluate the response to treatment. In our presentation, we are going to introduce you to the way microfluidic could improve the present cancer treatment, as the tool has proven to be performing complications on a microchip that solve these problems. A technology known as microfluidic uses tiny channels that range in size from 10 to hundreds of micrometers to manipulate small amount of liquid. These devices can provide rapid and sensitive assays for detecting specific molecules associated with cancer, allowing for early diagnosis and intervention. Before going into detail about the microfluidic system, I would like to briefly talk about the meaning of biomarkers, which will be mentioned in the next slide. Biomarkers serve as a measurable indicator of biological processes associated with the presence of cancer or specific characteristics of cancer cells. It is used in cancer screening programs to identify individuals at higher risk of developing cancer, and it can be used to classify cancer subtypes as well.
Microfluidic biosensors represent a promising technology in the field of cancer diagnosis. These biosensors operate by identifying specific biomarkers associated with cancer. At the core of a microfluidic biosensor is a microfluidic chip featuring channels and chambers to facilitate the flow of a sample through the device, allowing interaction with the biosensor. The biosensor is equipped with molecules that bind to cancer biomarkers. As the sample traverses the channels, the biomarkers bind to the functionalized biosensor, generating a detectable and quantifiable signal. This signal, whether optical or electrical, is then analyzed to ascertain the presence and concentration of cancer biomarkers, providing valuable information for cancer diagnosis and disease progression monitoring. For our optical sensors, they work by measuring changes in the intensity or wavelength of light that occurs when a biological molecule binds to a receptor on the surface of the sensor. For electrical sensors, they detect biomarkers by introducing a sample into a microfluidic channel, where biomarkers binding to the modified electrode induce a measurable change in the electrical signal, enabling the determination of their presence and concentration. A hypothetical Cas13-based microfluidic biosensor for cancer diagnosis is a device that uses microfluidic technology and Cas13 enzyme to detect cancer-associated RNA in a patient's blood sample. The blood sample is first collected and then introduced to the microfluidic chip. The nucleic acids are isolated and then followed into wells containing reagents for the detection of DNA mutations or quantifying overexpressed cancer-associated RNA. Cas13 is activated by direct targeting of RNA and subsequent trans cleavage activity, which triggers the catalytic hairpin assay and ultimately signal generation. The biosensor can detect multiple circulating RNA, including EGFR and TTF1 mRNA, along with many other. The designed chip can be used 37 times continuously for an RNA's detection without sensitivity reduction. In a study, the sensitivity and specificity of the biosensor for distinguishing non-cancerous groups from cancer groups with different stages were 97.3% and 95.2% respectively. This technology offers a precise, quick, and user-friendly technique for cancer diagnosis and treatment.
The advantages of the use of microfluidic in cancer treatment are: first, it can facilitate high-throughput screening of anti-cancer drugs for identifying personalized treatment strategies. Second, microfluidic devices can be adapted for point-of-care diagnostic, enabling rapid screening for cancer biomarkers, which leads to early detection of disease. Third, the device can potentially reduce the amounts of reagents, as it enables precise control over fluid flow and manipulation at the microscale, making the research process more cost-effective. Microfluidic is related to three main topics that we had studied. First, it is often considered as a subset of biomems, which refers to the integration of microscale and nanoscale technologies with biological entities, and microfluidics is a key component of this field as it involves the use of small-scale fluidic systems to perform various tasks related to cancer therapy. Second, microfluidic platforms are instrumental in designing and optimizing drug delivery systems for cancer therapy by allowing for precise control over fluid flow, and this is essential for designing drug delivery systems that can control the release rate and dosage. Lastly, the biocompatibility of the microfluidic devices are often used for cell culture. The materials used in these devices must be biocompatible to ensure cell viability and maintain the physiological relevance of the study. It is challenging to provide a specific average cost of microfluidic technologies in cancer treatment, as the expenses can vary widely depending on the use of each patient. But to estimate, the cost of microfluidic could range from $10,000 to $500,000.
While microfluidic systems for cancer diagnosis offer many advantages, they also have limitations. The first prioritized thing that should be considered is material limitations. PDMS, the most used material in microfluidic, has limitations that must be taken into account when developing the microfluidic system because it is related to various factors, and the noticeable ones are absorption and contamination. PDMS tend to absorb hydrophobic molecules, potentially altering experimental outcome and leading to inconsistent results. Contamination from the environmental due to the natural exacerbate this issue. Chemical compatibility. PDMS exhibit limited compatibility with certain solvents, acids, and base, risking device integrity and affecting the experimental conditions upon exposure. Mechanical weakness. Being soft and flexible, PDMS can deform under pressure, causing leakage and device failure, especially with high folate or small thin structure. Unscalability. Manual processing, long cruising time, limiting mechanical strength, and difficult in creating large monolithic structure make PDMS unstable for large-scale manufacturing. Anyway, I'm talking about production right now, and the manufacturing challenge also have limitation. High technology and sophisticated manufacturing process are required for micrometer scale structure. Mass production will be a challenge. About physiological complexity, current microfluidic device needs improvement to better recapitulate the physical complexity of miniaturization. Lastly, the application focus. Microfluidic system can be used in various applications. However, the current studies still have strong technical rather than an application focus, which makes the application in microfluidic system for cancer diagnosis not receive the priority as it should be. To address the limitation of microfluidic system for cancer diagnosis, several key improvements can be implemented. Firstly, enhancing the materials used in this system is crucial. This involves defining the material used such as glass, thermoplastic, elastomer, silicone, etc. Different types of materials contain their own key properties, which have different advantages and disadvantages. This leads to their usage in various applications, including cancer diagnosis, depending on the compatibility. Additionally, optimizing the design and functionality of microfluidic device is involved refining the microchannel geometries, enhancing fluid manipulation techniques, and incorporating advanced sensing. This technology can leading to improved accuracy and efficiency in cancer cell detection. Furthermore, manufacturing process is vital for mass production and cost reduction. Employing scalable fabrication techniques can help achieve consistent and cost-effective production of microfluidic device. Standardization of manufacturing process will not only enhance reproducibility but also contribute to the widespread adoption of the diagnostic tools. AI can enhance microfluidic system for cancer diagnosis by optimizing data analysis, pattern recognition, and decision making, thereby overcome human limitation. Although various components of microfluidic system for cancer and diagnosis are currently in use for sample processing related to cancer investigations, such as dark screen and delivery, cancer detection, and use in diverse approaches of treatment like gene therapy, chemotherapy, and radiation therapy. There is still a lot of room for improvements and developing overall system performance for more effective and reliable cancer detection. But if we can make the microfluidic system that is fully functional, this could revolutionize healthcare by enabling early detection, personalized treatment, and cost effectiveness, ultimately saving lives and reducing the burden of cancer on global health. Thank you.